Hello, in this video we will discuss about the jak-stat signaling pathway. So the genus kinase signal transducer and activators, basically the jak-stat signaling pathway. We will also learn about the other pathway which that is triggered from the jak-stat. So the genus kinase signal transducer and activator of transcription jak-stat pathway is a chain of interaction between protein in a cell. So the chain of interaction with the different protein. So let's begin to understand with the example. So there are many cells involved for this jak-stat signaling pathway, but here we can understand the lymphocyte. So here the dendritic cell, after the recognition of the antigen, which that is antigen representing cell, we represent the antigen through MHC class 2 molecule with the T lymphocyte. After the T lymphocyte will produce the inflammatory cytokines, which that will be bound with the other lymphocyte or other cells, and the tyrosine receptor is the basically receptor which that is used for the binding. So after the binding, the cascade of phosphorylation events will be occur, which that will become a transcriptional factor will bind the DNA to synthesize the protein or some other pro-inflammatory cytokines, which that can be bound with the other cell for proliferation. For example, the monocyte, which that will be bound with the tyrosine receptor with the inflammatory cytokines to proliferate into the macrophage. So that means the differentiation of the cell and cell division is very important for the tyrosine jak-stat signaling pathway. So in this way, here you can see this is the pro-inflammatory cytokine also bind the monocyte, for example, to go into the tunica intima during the atherosclerosis, or you can understand as well as the movement. So that is why the migration is very important, the migration of the cell. So the inflammation caused due to the pro-inflammatory cytokines, and inflammation is an inflammatory disease, for example rheumatoid arthritis. So let's begin to understand the whole mechanism with the receptor tyrosine bind with the jak-stat. So this is the dimer. This dimer receptor which that can be bind the ligand, can be hormone, cytokine or growth factors. After the binding with the growth factor or binding with a cytokine, for example, so the jak2, which that is phosphorylase, and a dimer, in this way the monomer formation, and the stat will be phosphorylase by the recruitment of the stat protein from the jak2, and after the phosphorylation will dimerization will be occur and dissociation with the receptor. After the dissociation with the receptor will go into the nucleus for transcription. So the transcriptional factor by the phosphorylation of the other protein also. So on the other hand, the other phosphorylation of the phosphoinositide 3 kinase, which that is phosphorylase, and the cascade of the phosphorylation with the PDK1 and AKT phosphorylation. So the on the other hand, the mtorc1 complex, the transcriptional factor AKT, will produce the mtorc1 gene activation to produce the mTOR. So this is the mTOR, and in this way here is the pi3k and AKT signaling pathway. This is the type which that is another pathway, and here is the stat pathway. This is the stat pathway. So this all things is very meaningful and used for the proliferation of the cell. And here on the other hand, the ip2 phosphoinositide diphosphate, which that will be activate in the presence of the pi3k phosphoinositide 3 kinase activation to lead to activate and phosphorylase the pip2 into the pip3. So in this way, the Ip3 phosphoinositide triphosphate with the phospholipase C enzyme used to break down, and the Ip3 will be inositide triphosphate will be separate. While here is the protein kinase C will be separate with the diacylglycerol. It will be recruit and this PKC is the substrate phosphorylation. So the pi3k phosphorylation and DDK1 again. So here is the Ip3 is the basically the Ip3 sensitive calcium channel are present in the endoplasmic reticulum will bind and the efflux of the calcium which that will be activate the protein kinase C, and this activated protein kinase C will bind with the diacylglycerol. After the binding with the diacylglycerol, the substrate phosphorylation will be occur. There are many other protein phosphorylase. So this all mechanism is used for the cell proliferation and cell survival. So on the other hand, here is the calcium. This is also used for the migration of cell contraction, migration of cell and the contraction inside that inside the microfilament and microtubule which that is used for. So the cytoskeleton movement, and this is the Ip3 pathway. And next is the here you can see this is the basically the RAS protein is bind with the cytoplasm cytosolic protein but intracellular domain here. So here is the intracellular domain attached with the GERB2 protein. So the growth binding protein 2, and the source will be bind after the recruitment, and the RAS will be activated into the GTP will activate the RAF, and RAF will be activate, and this activated RAF will be activate the MAC, this MAC phosphorylation, this protein, and after this protein phosphorylation the mapkinase complex will be activated, and this mapkinase complex is the transcriptional factor, and this is known as the mapkinase signaling pathway. So this mapkinase signaling pathway, which that is the cascade of phosphorylation events will be occur in this way. The IRF will be also bind. So here is the nucleus which that is contain DNA, and this DNA contain different genes. So this all protein will target as a transcriptional factor or target to the different genes. For example, here is the STAT. This STAT will trigger the further other genes transcription, for example. And here on the other hand, the mapkinase complex, which that will trigger the other genes, for example. So on the other hand, here is on the other hand we will discuss about the Mtor pathway, which that is the Mtor pathway will also stimulate to produce a different gene transcription for the translation to produce synthesis of protein. So we will discuss here is the STAT, a transcription factor will produce the RAS gene activation, GFAP and P21 and C-mice and CYCD, and here is the BL2, BCL2 and MCL1 and BCLXL and PIM. These all genes transcribe and translate into the cell proliferate differentiation through RAS protein and the GFAP. On the other hand, the P21 is a protein which that will be inhibit the cell cycle, while the proliferation, while on the other hand, basically the cell growth will be occur. And here is the cell cycle progression. So the cell cycle progression through C-mice. And on the other hand, there are these genes which that is used for the anti-apoptosis. So the cell will survive by these genes. On the other hand, the mtorc1 complex will trigger and will stimulate. Here is you can see after the transcribing to translate the mtorc1, which that is used for the cell cycle, cell survive and inhibit the autophagy. So that is why it is without the fasting during the fed state it can be active. On the other hand, the mapkinase which that will be is the promoter region bind with the promoter region to stimulate the cis and SOX. The cis and SOX will be inhibit the STAT and JAK pathway. So the STAT and JAK protein phosphorylation inhibition, because if the too much production of the STAT and JAK, so the inhibitory activity, so the negative feedback within the cell. So in this way, the here is after the inhibition will stimulate the ECS complex. After this ECS complex formation will ubiquitin mediated proteolysis will be occur, because the proteolysis is very important for the cell abnormal proliferation. So the apoptosis and autophagy will be occur. So in this all things the basically used for the cell proliferation on the range of the cell proliferation, because the excess of these gene transcription will not inhibit, so that means the cell will be progress into the cancer. So the abnormal cell division will be occur. So that is why the cell cycle inhibition through P21 gene, and on the other hand the cell cycle progression will also, which that is all things will be inhibit and stimulate to continue regulation of the cell division and cell proliferation. So here the cancer progression, for example, will be occur if the migration, proliferation and inhibition and mitosis, meiosis and growth and anti-apoptotic action, and all these things will lead to promote the survival while whole body normally. If our expression of the gene can cause cancer, so the proliferation of the our expression of that genes can cause cancer, but it is normally work for the normal physiology in our body. So I hope you are getting this concept about the jak-stat signaling pathway. So thanks for watching. Please make sure to subscribe, like and share. Bye.